Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
This is Richard Jacobs with the Finding Genius Podcast. My goal is to find exceptional individuals that I can ask questions of, and they'll reveal things that you may not see in articles, that have done about them, in videos they've been in. Ask them questions where they say, "Oh, that's a good question," and then I know we're getting to the good material. So that's my goal here. My guest is Brian McKelligon. He's the CEO of Akoya Biosciences. It's Akoya. We're going to be talking about what's called Immuno-Oncology. So I guess that's the interplay of the immune system as it relates to cancer, but he'll get into that. So, Brian, thanks for coming.
Thank you, Rich. Really appreciate your time.
Yeah, so if you don't mind, what's the mission of Akoya Biosciences?
So Akoya, as a company, our real mission here is to help those scientists that are working in academic, biopharma, or government research institutions to better understand the underpinnings and the pathophysiology of disease. And there's a lot of technologies that are deployed to do that. And we can talk later about what's unique about our technology, but that is our overarching mission. Much of our focus, given the nature of the technologies that we deploy, is really within this immuno-oncology field, this exploding therapeutic modality that treats cancer by really unlocking the power of your immune system. And inherent in developing therapies, new therapies, advancing therapies in that realm, inherent in the development of those therapeutics is a need to really understand the most relevant biomarkers, and not just whether or not those proteins, those RNA molecules, or those mutations are present. Not yes or no, are they present, or how much, but actually where in the tissue they're present. Is it inside the tumor? Is it in the surrounding stroma? And so our mission is to really deploy technologies that provide sort of next generation tissue analysis for those researchers working in those fields where those questions are paramount. So that's kind of an overarching summary of what we're trying to do is really serve those scientists and their needs, again, with a lot of focus in this field of immunology.
I understand that cancers like to hide from the immune system. Somehow they're able to disguise themselves. So immuno-oncology seeks to retrain the immune system so that it does recognize cancers and gets rid of them. Is that right?
Well, there's certain proteins and certain pathways you may have heard about, and these therapies that interrupt this checkpoint cascade, this ability for cancer to hide in plain sight. What these therapies are doing is they're shutting down those pathways, PD-1, PD-L1, and allowing the introduced therapies to immediately recognize the presence of cancer as a foreign entity and then begin to attack it. And so what you're seeing is just this explosion of clinical trials. Maybe it's one or two new trials a day that are really these combination therapies between existing drugs, existing antibodies on the market, monoclonal therapies, and new ones, trying to come up with the right combinations that shut down this checkpoint cascade that allows these cancers to effectively hide in plain sight. And in doing so, it's really revolutionizing how we're treating cancer, again, by leveraging the power of your own immune system, rather than treating you with pretty harsh therapeutic methodologies.
What are some of the trade-offs or the difficulties in activating the immune system to fight cancer? What's so hard about it?
Well, I think the hard thing is that, you and I, our bodies might recognize and present, different proteins that our body might recognize as foreign. There's a lot of challenges in coming up with, and I don't want to pretend to be a physician rather than a sort of a technologist, Akoya is, but one of the reasons why platforms like ours are gaining an increased presence is while these therapies are incredibly powerful, and you can, some of the names you might recognize, Keytruda and Opdivo from Merck and BMS. These are incredibly powerful therapies when they work, and when they don't work, it's incredibly expensive, and you sacrifice another therapeutic path. And so the challenge we have right now with these immunotherapies is the ability to predict whether or not a patient can respond is a little bit underwhelming right now. And companies like Agilent and Ventana have done an incredible job of coming out with biomarker technologies that predict whether or not a patient will respond, but they're just not powerful enough. If I test you for one of these biomarkers, PD-L1 for example, and you're positive for it, your chances of responding are maybe 30 or 40%. If you're negative to that biomarker, indicating that you're not going to respond, your chances of responding are still 10%. So the challenge we have is that these therapies are incredibly expensive. And so coming up with more predictive biomarkers would be an incredible boon for the patients, for the health economics, and for the continued advancement of the field. So we're working with our customers to try to better understand that tumor environment, that tumor microenvironment, to help them come up with more predictive biomarkers. So if you walk in the door at a Memorial Sloan or a Dana-Farber or UT, and they're going to run a biomarker test to potentially put you on one of these powerful therapies, that it's a lot more predictive, and that you're making the right therapeutic decision with your physician.
There's at least hundreds of biomarkers. How do you know where to look? Is there a tool that guides you or some kind of heuristic?
Yeah, I mean, so, I think what you're seeing in the market is suites of technologies. I'd like to sit here and say that Akoya is the only thing that people need to use. But what you're seeing are suites of technologies that people are deploying to try to better understand which proteins, which DNA mutations, which RNAs are expressed. What are the technologies that are most predictive? And generally on this sort of biomarker development continuum, to simplify it, you have a couple of phases. Phase one is truly exploratory. And with that, you need to look at as many markers as possible. So you might take a series of tumor samples and run whole genome sequencing on them. You might do single cell with a Illumina or an ion torrent Thermo Fisher. You might do single RNA analysis using something like a company called 10x Genomics in Northern California. And then where Akoya steps in is looking at the protein. And so what we try to deploy for those scientists are solutions that allow you to look at a tissue and not look at one or two biomarkers, but look at 30 or 40 or 50 standard technologies to analyze a tissue. If you go into a doctor right now and they take a biopsy and they want to look at a protein marker, usually 1, 2, maybe 3 biomarkers you can look at the same time. Maybe just to pause for a minute on my storyline, just to give a little bit of background. So the technologies that are deployed for histology, looking at a biopsy, looking at a tissue sample on a slide, historically 1, 2, 3 proteins at a time. So we can sequence your genome for under $1,000, which is amazing, but we're still using technology that's 20, 30 years old to look at tissue samples. And so what Akoya brings to the market are next generation tools to look at these tissues. So immunohistochemistry is using immunoantibodies to look at cells on a tissue. And so what Akoya is deploying as part of this exploratory biomarker phase is a technology that came out of Dr. Gary Nolan's lab at Stanford called Codex. And this allows you to look at 30 or 40 or 50 different proteins on a single tissue slide and maintain that tissue integrity, so you not only know where those proteins are present, but how much. So that is a discovery phase of biomarker development. And then once you know the key biomarkers you want to look at, now you're going to pivot to look at a large population, a larger clinical study. Maybe it's hundreds to thousands of samples. And for that, you really need a high throughput engine. And that's where one of our other techniques steps in to really do this. So
it's been noticed when you look at a tissue, even the ones you've looked at, and you see the patterning of various proteins and the biomarker expression of the tissue. I mean, I'm sure new things have been noticed that weren't noticed before. Do you know what they are?
Yeah, so there's some interesting things that are getting revealed at a recent conference at the end of last year, one of our partners presented a really powerful study looking at melanomas, identifying a number of proteins that together, when you look at their level of expression, low, medium, high to simplify, and their location, provide hope that we'll be able to predict outcome of therapy using these 6 or 7 biomarkers. Before, we weren't able to really predict one's outcome. And so what this affords us to do, again, there's a lot more work to be done, but what our colleagues Drew Pardoll and Janice Tao presented at a conference last November, I believe, are really some interesting studies that look at these biomarkers, their presence, their expression levels, and their location, and provided some really interesting insights on our ability to predict whether or not they're going to respond to therapy. And that's what's needed in the market with these IO therapies.
Any sense of how predictive what you're seeing will be?
No, not yet. I mean, we have indications, but I think we'll leave that to our scientists at Hopkins to get to that level of conclusion. I think there's a number of avenues right now in melanoma, in breast cancer, in dermatological disease. Certainly the coronavirus. The technologies that Akoya is deploying to market are fairly new and novel, just a couple of years old, and so we're just beginning to scratch the surface of the power of what we call spatial biology, and that's what this is. It's a new concept in biomarkers that contemplates biomarker presence and location, and so we're getting really powerful indications, and a number of publications. 2 just came out over the last 2 days in transplant medicine around the power of these biomarkers and their location in tissue as being predictive. So we're not yet at a point, Rich, where these are diagnostics. Obviously, that's our longer term aim, to continue to press our technology from exploring novel biomarkers using this concept of spatial biology to then further testing them in larger clinical samples, to then deploying them in diagnostics. So we're really in those first 1 or 2 phases right now, Rich, not really yet at the realm of full diagnostics. So I think that's the hope for the company. When we went out and sought additional funding at this time last year, that's really a key to our story, is that Akoya is marching down that with our partners, and that's one of the reasons that that Agilent, I hope, invested.
Find that biomarker concentration of something falls off as you get away from the center of a tumor. What does that tell you? If it has a certain directionality to it or a change in concentration, like what have you seen so far that you can comment on, and what does that seem to be telling you, even though it hasn't been proven yet?
So what's important is that you need a protein and its receptor. You need the right cell types present within the tumor environment, and not necessarily measuring it in the surrounding stroma. So really, that's really at the core, that is identifying the relevant markers and coming up with computable spatial metrics that give you a sense that, yes, these markers are present. That's the underlying biology. Therefore, it needs to be in this location, proximal to these other cell types. So one of our partners, as an example, that's working in breast cancer and trying to really change the mode, and to oversimplify a bit, just trying to change the standard of care for breast cancer. They've got, I think, something like 15 different markers that they're looking at within a breast cancer biopsy. And with those markers, they have, dozens of proximal measurements of this marker and its distance to another relevant marker because of the biology. And so they come up with this fairly complex framework of computable distant measurements that in aggregate have some predictive power. And that I think the future of spatial biology in the biomarker market. So if you look, for example, historically at what revolutionary companies like Genomic Health, now part of Exact Sciences, have done, is they came up with a method to look at 25 different RNA markers and have something called a recurrence score that's going to predict your response to certain cancer drugs. And that was a computable metric based on RNA expression. And I think where we're going by comparison, we hope, is getting to a point where the computed metrics are now distance measurements between cell types and the relevant biomarkers that can predict that some underlying biology is happening, like your body is or can respond to a tumor via a catalyst via an immune therapy. So that's kind of a general sweeping summary of how we think about biomarkers within the spatial biology context.
What I'm imagining is you'll look at, I don't know, it may be spatial, it may not, but let's say you look at 30 biomarkers, you'll probably be able to establish in these patients there's a predominance of markers 1, 6, 9, and 13, and in these other patients there's a predominance of 2, 4, 8, and 12. Therefore, it'll create like clinical profiles that you'll treat differently based on not only the spacing, but again, the predominance and the concentration of certain biomarkers. And that probably would work a lot better in terms of treatment instead of just one, shotgun blast of whatever drug it is.
Yeah, I mean, I think that's the hope. And if you take one step upstream towards the development of those biomarkers, what you need, well, so just back up. Imagine that I've got melanoma, and they take a biopsy, and that biopsy then gets thinly sliced and placed on. That's what happens. Now, if you look at that slide under the microscope, pathologists with their expert training can identify cell type. But if you're in a drug discovery environment, or you're a professor in a research institute, and you're trying to understand the evolution of cancer and how certain cancers like melanoma evolve over time, and perhaps you were trained as a cell biologist or in a field that's not pathology, you're not going to have that expert training to be able to understand visually by looking under the microscope all these different cell types. So there's also kind of a fundamental accelerant in terms of enabling these sort of immunohistochemistry, immunofluorescence to be used by scientists that are not expertly trained pathologists, because we can come up with biomarkers that identify a T cell, they identify the B cell, they identify the macrophages, they identify the endothelial cells. We can come up with markers that identify all the cell types, and through our software, we can label those cells. That's a B cell, that's a T cell. And so now what you have, and this is actually what our software does, now what you have is a slide that's been labeled with the actual cell types, so you can begin to visually and computationally make predictive decisions across these cohort of samples, which are the most predictive biomarkers and what's happening to the underlying cancer biology. So in addition to identifying the predictive biomarkers, which you appropriately described, what's also needed are some of these fundamentals so that technologies like ours, like immunohistochemistry, like looking at tissue samples, can be broadly deployed, and that power can be unlocked to a broader suite of scientists. And I think that's what's making these sorts of spatial biology solutions, increasingly powerful.
Thing that, I know you can't look at everything. One thing I don't hear a lot is a reference to the microbiome and the microbial attachment that it looks like is next to all tissue. For instance, I interviewed a lady, Florencia McAllister, that looks at pancreatic tumors, and she's saying that not only does the pancreas have a microbiome, but the tumors themselves have their own unique microbiome. So when you're doing histology on certain tissue, I don't know if you're able to see if there's any microbes there, because what if the microbes are responsible for the predominance of a certain biomarker or not? What if they're interspersed within the tissue, or there's a biofilm on the tissue or in the tissue that changes what is there? I don't even know if it's being looked for, and that may be very important.
Yeah, so I think, in short, are we working with customers right now to ask and answer that question? I'd say not broadly, but our degree of excitement in leveraging our platform and working with scientists to address questions like that, obviously that's where we want to go. We don't yet, I think, have a lot of data that we can deploy to answer the specific microbiome question, but that's absolutely true. And one of the founders, the founder of one of our technologies I mentioned before, Dr. Gary Nolan, he's got some work in his lab where they're looking at you leverage our technology to look at certainly infectious disease, and now more recently there's some tweets you'll see from him looking at COVID. So I think certainly the technology can be deployed to do that, but not to the extent yet.
The point is that you're making is that your analysis and your histology will be so much better than what's out there. It should help tremendously, right?
I mean, yeah, I think our basic point here is that to draw a poor analogy, since we're all stay at home these days, our our cellular environment doesn't work like we act right now, sitting in our houses by ourselves. There's interactions happening on a single cell level that, in order to appropriately measure them, you have to measure the expression and presence of these proteins and of the transcripts while maintaining that tissue integrity, like I said. So you know not only what is present, but where it's present. And that really is now the third axis of biomarkers, presence, abundance, and location. That's really been the evolution of this term spatial biology. That's why it's increasingly important. And I think that over time, it's just going to continue to explode, this triple axis of biomarker discovery and diagnostic.
So you said Akoya is in the early stages. What's the roadmap look like for the next few years? How long till this analysis would appear in clinical use?
So we've got a number of partners at large academic institutions that are deploying this across large scale clinical trial. We have our technologies that are broadly deployed farther upstream in the discovery environment. So it's already being utilized within the clinical trial environment. So at what point do you think, you or I could walk into a physician's office and our technology will be as part of formal diagnostic use? I think that's still a few years old.
You're on the path, so that's great. So what specific types of conditions do you think this will be most amenable to? Are there certain cancers like melanoma you mentioned a few times, or is that just your focus, or do you think this will be useful for any type of cancer?
So generally, the underlying technology for our spatial biology solution, multiplex immunofluorescence, multi meaning a lot, immuno meaning antibodies to look at proteins in tissue, and fluorescence meaning that's how we detect it. So multiplex immunofluorescence. I'd say that the tip of the spear for us in terms of how it's being deployed is really within solid tumors where these immuno-oncology therapies is the modality. So that lung, melanoma, I think right now there's about 14 different cancers, breast cancer. So that's really the tip of the spear. But we're seeing the deployment of our solutions getting wider and wider with each passing month and quarter. It certainly has applicability, as I said, to dermatological diseases, in transplant medicine, where immunoresponse, similar to immuno-oncology, is really relevant. I think the applications in the neurological space are profound. We have a number of customers using it that way. And then over time, I think you'll see it as cell therapies begin to explode onto the market, particularly those that are coming out of stem cell therapies, where you really want to measure not only the cellular state, because we're differentiating these cells, but also whether or not they're getting taken up by tissue and the response they're having. So there's a number of different therapy areas where we have relevance. But to your question, immuno-oncology, lung, melanoma, I think those are really the tips of the spear.
What's the best way for people to find out more and keep tabs on Akoya and what you're doing?
Yeah, I think the best way is to go to AkoyaBiosciences.com and take a look at our web. We try to do a blog on a regular basis. It provides some good context around how people are using our technology and areas it's being deployed. I think that's probably the best way.
Any final words that you want to say about this?
I appreciate your time, and I think the final words is that the technologists and the scientists that are coming up with revolutionary therapies like the immuno-oncology drugs we've talked about, equally need technologies to identify the relevant biomarkers to further catalyze that work. And ultimately, that's what we're trying to do is help those scientists to help patients. And that's our goal as a company. And if we keep our eye on that, we'll be successful.
Ryan McKelligon, CEO of Akoya Biosciences, thank you for coming.
All right, Rich. Appreciate it. Stay well.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.